So let's take a look at vision and the testing of vision responses. First, we have the external anatomy of the eye. Note the various structures that happen to be there. Note the central location of the iris and the pupil within the eye. This central location is important to keep visual center within both eyes. Note that within the eye there are two distinct cavities. The two distinct cavities are referred to as the anterior cavity and the posterior cavity. The anterior cavity is going to reside between the cornea and the lens and is made up of two chambers, the anterior chamber and the posterior chamber, which is separated by the scleral muscles of the iris. At the lateral border of this anterior cavity is a sinus, the scleral venous sinus. This sinus is what gets blocked when someone has the disease known as glaucoma. This blockage leads to an increase of fluid within the anterior cavity. This fluid buildup within the anterior cavity causes excessive pressure, and it's that excessive pressure that causes all of the visual issues for the person with glaucoma. Note the orientation of the ciliary body and the suspensory ligaments onto the lens. These ciliary bodies and suspensory ligaments will keep the lens in the central location of the eye, as well as correct for curvature when accommodation is necessary. So let's take a look at the anatomy of the eye. Once again, note all of the various structures. Note that there are external or extraocular muscles, referred to here as the superior muscle. We have the superior rectus muscle, the inferior rectus muscle, as well as oblique muscles. Note we also have muscles that control the opening and closing of the eyelids, usually referred to as the papillary muscles. These muscles control the movement of the eye in an attempt to keep the iris and the pupil centered on what we are visually attempting to look at. As we work into the internal anatomy of the eye, note that there is what's known as a visual axis. This visual axis is the direct orientation of light going onto what is referred to as the fovea. The fovea is the central portion of the retina where we have the highest density of cone cells and the greatest visual acuity. Once again, notice all of the extraocular muscles and their orientation. So let's take a look at the retina itself. The organization of the retina leads to a distribution of cones that are most dense at the fovea and least dense near the nasal or temporal margins, and where rods are most dense at the nasal and temporal margins and least dense at the fovea. This unique organization for each person leads to a differential response to peripheral as well as central representation of the images. We also get a distribution of cells within the retina, where we have an organization that goes from deep to superficial, where deep is closest to the pigment layer and superficial is closest to the posterior chamber or the vitreous humerus within the eye. This organization has the photoreceptors deepest and the neural cells most superficial. This attempts to diminish the amount of energy stimulus striking the photoreceptors so that the photoreceptors do not become overstimulated. Please note there is no true desensitization or attenuation, something we'll talk about in a few seconds here, to the photoreceptors. Photoreceptors also work off of an on-center, off-center organization with interaction to the bipolar cells, as well as a lateral inhibition from the horizontal and the amacrine cells. What this does is allows for distinct visual representation of the external environment on the retina. Within the retina, there are two distinct types of photoreceptors. The first type is what are known as the cone cells. There are three distinct types of cone cells. Each one will have its own distribution within the retina and will be distinctly responsive to various wavelengths of light for maximal excitation. These include the short wavelength cells, which we call the blue cones, the medium wavelength cells, which we call the green cones, and the long wavelength cells, which we call the red cones. All cone cells use an idopsin or a photopsin that is specific to that wavelength. So the blue cones would use blue photopsin, the green cones would use green photopsin, and the red cones would use red photopsin. Please note, if there is overstimulation to any one of the individual cone cells, we get a temporary desensitization where when we see colors, as in white light or normal light, we will not actually perceive that distinct color until the cone is able to be responsive again, meaning it's dissipated the overstimulation energy. There is no attenuation on the pathway. The other type is the rod. The rod has a one single distinct cell that is generalized to various lights, but needs a distinct amount of energy impart on it to get threshold in order to fire and send the action potential. The rods are what we usually use for low energy stimulus, such as what we see at night, and utilizes what's known as a rhodopsin for its pigment within the photoreceptor itself. Additionally, we break up the retina into distinct halves, where the inner half is referred to as the nasilar portion, and the lateral part is referred to as the temporal portion. This division is based off of where the actual sensory information is coming from the external environment in what's known as the visual field. Not only that, we get a crossover of information within the progression of cranial nerve 2, the optic nerve, from the retina through the lateral geniculate nucleus and then into the primary visual cortex. Within the retina, we get a topographic organization that's going to correspond to the distinct type of cell as well as the distinct location within the retina that we had stimulus. There's a secondary thing that occurs within this visual field, and that's a pathway that goes from the lateral geniculate nucleus through the pulvar nucleus and into the amygdala. This pathway here is for instantaneous visual response, where we do not actually have to process this information. Within the pulvar nucleus and within the amygdala, we have neurons that are set up to distinguish distinct patterns of stimulation so that we have instantaneous response. These include images such as what we see with snakes and what we see with faces. Within the projections coming away from the retina, we have two distinct branches of information being passed. The two distinct branches of information being passed are referred to as the temporal and the nasilar. The nasilar pathway is going to cross, it's going to desiccate to the opposite or contralateral side of the cerebral cortex. This occurs at the optic chiasm, and it junctions and joins with the ipsilateral side, the same side as the eye. Note the blue and note the pink. What these then do is then spread into what are referred to as the optic radiations, leaving the lateral geniculate nucleus and heading into what's known as the striate cortex, or the primary visual cortex. Additionally, there is mapping that occurs within the lateral geniculate nucleus that is based off of the organization of what's known as simple cell, convergence, and divergence that leads to the hypercomplex cells within the primary visual cortex. What this does is allows for communication so that we can actually establish a three-dimensional representation of what goes on within the external environment for both color as well as depth, as well as what's known as binocular communication, meaning the ability to perceive from both sets of eyes. If we do not get perception from both sets of eyes, we minimize the amount of depth perception that we have. You can see this by simply trying to look through your left eye and then look through your right eye, and you will notice that the image will change based on what's known as a parallax. The organization coming away from the parvocellular and magnocellular regions within the lateral geniculate nucleus gives a generalized organization of information heading into the primary visual cortex. What this does is this will establish organization based off of whether it's a rod or a cone being stimulated. What this organization does is it will spread from the primary visual cortex into secondary and then tertiary visual cortexes, where once we get to the V3, V4, and V7 regions of the visual cortex, we can then actually get full three-dimensional representation of the image that we are looking at. There's an interesting phenomenon that occurs with light. Light always travels in a straight line. Because light always travels in a straight line, it will be refracted and reflected based off of interacting with either a change in median or a change in surface, which means that light that strikes the right side of the lens in the cornea or the inferior side of the lens in the cornea will be bent to the opposite end of the retina. The same thing occurs for light striking the left or the superior. However, light passing straight through the center will continue to pass through the center. What this means is that the reception of information at the retina is inverted and reciprocated, meaning that it's flipped upside down and reversed left to right from what is actually seen in the environment. This pathway will continue through cranial nerve 2 until we reach the lateral geniculate nucleus, at which point in time the organization will be flipped so that the neurons in the primary visual cortex will be receiving stimulus that will match what occurs within the external environment. So let's take a look at how this occurs within the visual pathways. So if we look at the right and the left eye, we have two distinct receptions within the retina coming from either the temporal half or the nasilar half. This information will then be passed. Once again, the information striking on the nasal side will be converted and transferred to the opposite side of the cerebral cortex, whereas the information being received on the temporal side will stay on that side of the cerebral cortex. But note that the temporal retina will be picking up information coming from the nasilar side of the visual field, whereas the nasilar retina will be picking up information from the temporal side of the visual field. Now if we look at it in terms of what's occurring with the information coming through we have light striking. This light strike gets bent on the retina. That organization continues through the geniculate and the tectile plates, at which point in time the organization of the axons leaving via the optic radiations will revert to the initial state. So what is actually perceived in the visual cortex matches the external environment. However, what is received at the retina is inverted and reciprocated from the external environment, something we'll see in the next slide. So if we take a look at what's going on with the two images here, looking at the tree and looking at the pintac, if you notice, the reception at the retina for both images have been flipped, have been inverted and reciprocated from what is seen in the external environment. The way in which we can get the images focused onto the fovea is based off of what's known as accommodation to near and far point. Accommodation is strictly talking about the change in the curvature of the lens relative to the distance at which the object is to the retina that allows for maximal bent of the light to the fovea. Near point is going to minimize, whereas far point is going to maximize. And it's all about changing the curvature of the lens by increasing or decreasing the tautness of the suspensory ligaments coming away from the ciliary bodies. There's other issues at play here in terms of the distance from the retina to the lens. And this has to do with what's usually referred to as near sight and far sight. Here is normal. Here is near sight. Here is far sight. On the near sight, there is a diminishment of the superior inferior axis and an increase in the anterior posterior axis of the eye, which pushes the retina further away from the lens. For the nearsighted person, we correct this by adding a concave lens in front of the lens of the eye, which causes a refraction away from the central point, allowing for proper bending of the light onto the retina. Whereas in the farsighted or the myopic eye, the hyperoptic eye, there is an increase in the superior inferior axis and a diminishment in the anterior posterior axis. In this case, the retina is actually moved closer to the lens. We correct this by adding a convex lens in front of the lens of the eye, which causes initial refraction towards the center point, allowing for focusing of the light onto the fovea.